I want to use this opportunity to give a sort of perspective in general, how you can think of biology can contribute to the questions of climate and energy challenges that we arise, and where there are opportunities for biology to make an impact, and which areas are not the fortune of biology. So I want to use the first few minutes just to outline general how biology works and how you can think about it and make use of it, and then get into a microbial electrosynthesis. As Arun has pointed out, the big part here, that is the biggest symbol here, is GSAP. And I really want to explicitly thank GSAP in the early days when GSAP started to fund our research on microbial electrosynthesis. When it was truly in its infant, there was no promise that this technology would work out. We had to understand the basics, and I really appreciate the continuous support of GSAP to really get this technology going. And I hope I can convince you that actually this is probably the most viable technology in terms of biology to make an impact on climate and energy technologies. So what is the typical use of biology today in energy sciences? Well, biology has been used here to produce bioenergy crops. And bioenergy crops, you have a number of diverse crops that are then being used ultimately in large-scale ethanol plants to produce ethanol or butanol or other types of second-generation biofuels. Another approach that has been popular for a while and is still pursued by a number of industries are algae biomass, where ultimately also the energy from light that is used here to produce land crops is here to produce algae biomass. And this bioalgae mass is then processed typically to produce some kind of diesels. Now, if you think about these technologies, the question is, on what aspect of biology are these technologies capturing on, and what can you expect maximally from this type of biology? This is the secret of biology, and if you take out one thing from this lecture today, it's basically this way to think about biology. This rectangular here is a cell, a microbial cell. And microbial cells, as well as plant cells, do stuff. And the stuff that they do is basically metabolism. Now, there's a red and a blue arrow going in, meaning that the cells are utilizing some substrates and producing some end products. And ecologically important, the end products are offsprings, just more cells. So most of the elements and the elements go essentially in producing these offsprings. These biochemical reactions that take place in every cell, in bacterial cell, in a plant cell, also in our human cells, is called anabolism, because you're putting stuff together. The driving force for that, the thermodynamic driver of that, is energy in the form of ATP that is delivered by a second set of biochemical reactions. And these are called catabolic reactions. They are qualitatively different from anabolic reactions in the sense that these reactions are often faster and can turn over more substrates into products. The second implication of that is the end products of this metabolism, that is abbreviated here as P, is typically released to the environment. So for example, when we eat glucose and oxygen for breakfast or for lunch, we have CO2 and water that comes out. The energy is then used to produce biomass. Now, in order for this to proceed, the free energy change of this chemical reaction has to be negative. That has to be an exergonic reaction. How exergonic that reaction is has a huge impact of the flux of matter through a microbial cell or any plant cell or animal cell for that matter. Because we're putting, let's say, organic matter here that flows into this catabolism and also that flows into this anabolism, and you as sort of an engineer or any person who wants to use biology, you care how much of your substrate ends up either in cells or in the products. Now, with products, you can do a lot of stuff. If you produce primarily biomass, you have a chemically heterogeneous system. You have amino acids, you have lipids, you have nucleic acids, you have fatty acids, on and so forth. So chemically speaking, it's heterogeneous. The end products typically are uniform. That's the value. Now, what is depicted here is there is an intrinsic function of the delta G of this reaction. In other words, how exergonic this catabolism, these faster reactions are, depends on the amount of energy that is released. In other words, if there are small amounts of energy released, like in many anaerobic or microorganisms, then more of your substrate flows through this catabolism and ends up in the product, as opposed to a sort of highly exergonic reaction, which releases a lot of energy, like when we eat our breakfast, then most of the substrate actually goes to make biomass. So if you want to use biology, then you have to ask the question, am I interested in producing a lot of biomass, which I then can utilize and extract the compounds of interest? For example, if you have algae biofuels or so, we are talking essentially about lipids that are produced and embedded here in cells. And for the efficiency of this process, I need to think about what fraction of my original substrate actually flows via this arrow as opposed to that arrow. On the other hand, if the energy is small, like in anaerobic bacteria, for example, those that ferment glucose to ethanol, more, in fact, up to 90% of the substrate is actually converted into products, and it's pure. So therefore, depending on what process you make use of for your bioenergy systems, you have different efficiencies of where your substrate goes. And here are some examples of that. As I pointed out, when we're talking about algae biomass, so you grow algae, the sunlight is free, but you need nutrients that ultimately form this heterogeneous biomass. We are talking about anabolic fuels, because this is really what you want. So you're essentially pushing carbon, CO2, in this direction. And although some algae biosystems can accumulate up to 70% lipids, it is still a fraction of the total biomass that has to be extracted. So while the number sounds large, in reality, it's actually a small fraction of all the substrate that flows through this. Another example is if you take plant biomass. In plant biomass, you have also the energy coming from light that is used to provide the energy to make cells, or basically the biomass. If you ask the question, how efficient is the biology, and count 100% of the light that shines on the surface of a plant, you get only 1% or less than 1% out as chemically usable energy. And that has to do with how efficient light photosynthesis is, so the light harvesting, how efficient the conversion of this photo energy to chemical energy is, how this energy is split between anabolic and catabolic reactions, how this energy is used to deal with ecological systems when you have diseases, you have storms, we have inefficiencies. So if you take this less than 1% of biomass energy and then make something useful out of this, like hydrogen, this is cut back by another factor of 8, so you're ending up with something like 0.2%. So biomass itself has sort of challenges if you want to use it as a system for providing solutions for energy. Biomass has been used successfully by making use of this principle here in bioethanol farms, like here, the one in Iowa, where in a catabolic process, so you have high rates and an efficient funneling of your substrate to an end product. So that's good. you produce ethanol, and the organisms are resistant to molar concentrations of ethanol. So the only reason why good red wine has not more than 16% is the yeast is dying. But if you translate this to molar concentrations, it's like 1.5 molar. So for those that know chemistry, that's a lot. But the organisms are tolerant to that. So in that sense, the anabolic fuel, and I call this ethanol anabolic fuel because the substrate that goes in is actually an anabolic substrate. It's plant biomass. It's heterogeneous plant biomass. So there are intrinsic limitations to that, how you use essentially biology in this system. Now, in order to sort of transit to the microbial electrosynthesis, I want to work out one fundamentals of these catabolic processes. And that is actually what is behind this bar where organisms in their energy metabolism, which is a fairly efficient, chemically very selective process, what are the rules of that game? Well, the rules of that game was nicely summarized by Alfred Spormann, who discovered vitamin C, who said, life is nothing but an electron looking for a place to rest. So what does this mean? There are two levels of important information in there. First of all, life is redox chemistry. So it's flow of electrons through a biological system. And putting an electron to rest means it's going thermodynamically downhill. So specifically for any biological system, what that means is that this S here, the substrate, the substrate that can be efficiently converted into an end product, is actually two substrates. It's a substrate 1 and a substrate 2, where in this process, in this biochemical machinery, substrate 1 becomes oxidized, so you remove what the biologists say here as a hydrogen atom, which is nothing else than electrons. And these electrons are then transferred to a second substrate to produce an end product. For example, when you eat breakfast, your cereals, glucose is the substrate 1 that becomes oxidized to CO2, and oxygen is the substrate 2 that becomes reduced to water. That's intrinsic to all forms of life, in particular to microbial system. In fact, photosynthesis is just a convoluted way to get an electron flow going. So when we're talking about respiration, this is all lumped together here. It's just a convoluted way to get an electron flow going in order to conserve energy. So therefore, microbial life depends on oxidation and reduction reaction. And all biotechnology makes use of soluble substrates. So you have essentially glucose, you have acetate, you have formate, you can name it. It's all soluble that you put in a fermenter, and then you have an electron acceptor, and the organisms produce end products which are typically desirable, unless you want to do biomass, but then there's a high cost associated for that. So our approach was now to ask the question, how does a microbial system behave if the electron donor here, the substrate, is not soluble but insoluble, and actually if it's a cathode? So what this basically means in the overall scheme, this is what we are doing. We have our microbial cell, and we are using cells where you feed electrons in not from a soluble source, but from a cathode. And we use CO2 as electron acceptor. And for the organisms to make a good living, essentially conserve energy, they are producing methane, natural gas, C2 carbons, C4 and C6 carbons. In other words, you can produce multi-carbon compounds at high selectivity and at high rate and at high efficiency from CO2. The implication of that is this technology then allows you to use renewable energy and ultimately store renewable energy in form of chemicals, like methane is a great energy storage compound. And it also produces, you can produce other fuels or other precursors for chemical syntheses that are ultimately derived from CO2. So the utilization of that methane is good because this methane is green. The other advantage is we are working with organisms that have a small delta G. So the change in free energy is small. That means we have a high yield of converting the total energy into the end product. And if we're looking at the Coulombic efficiency of this process, we know from our data it's more than 90%. So how does this work? This is our microbial electrofuels reactor, where we're taking electrons from water or from waste, energy from renewables, or nuclear for that matter, which is then converted into useful compounds based on carbon, based on CO2, that can be used. And then the CO2 that is produced there is basically also green in the sense that it's no net CO2 introduction. And you essentially can have an economy based on this cycle. Practically in the lab, that looks like this. Sort of, it's a highly wired system where we have microbial reactors. So the light doesn't show it that well, but that's all right. Where we have here a cathodic chamber where we control the potential with a reference electrode. And we have microorganisms in there. And then here we have an anodic chamber separated by an ion exchange membrane. Here we have a variation where we have a 3-compartment chamber with the microbial system in the middle. The question now, if you want to use this technology, is what is the mechanism of uptake of electrons from a cathodic surface into bacteria? Because that's something that is unclear, because it's not in transporter that you would find normally. Now, in as exotic it may sound, nature has been doing this already. And there were 2 reports that are really worth mentioning. One is a discovery that a Japanese group published in 2010 at deep sea hydrothermal vents. So deep sea hydrothermal vents at the tectonic plates in the Pacific and the oceans, you have deep sea hydrothermal vents where highly reduced hot fluid, typically reduced containing hydrogen and sulfidic minerals and sulfides, escape into the seawater and can reduce the chalcopyrite, which is essentially an iron sulfide, which is electrically conducting, push electrons into this mineral phase, and outside in the cold seawater, these electrons, derived ultimately from sulfide, can be oxidized microbially with oxygen. So here we basically have essentially microbes oxidizing a cathode, and this cathode in this case is actually a mineral surface. Another system where we have a microbial consortia that degrade ethanol to methane, the group has shown that this occurs via a direct contact between these ethanol oxidizing bacteria and methanogenic organisms that actually can take up these electrons by an unknown mechanism. So in other words, while the idea of using a cathode as electron acceptor is artificial, nature has several systems already that have the fundamental biology to that. So how does it work? It started in 2009 when Bruce Logan, who had also a GSAP grant with us, made the seminal discovery that in a mixed culture system, electricity on a cathode could be converted to methane. And methane is an ideal system because it's a gas that is soluble at a low concentration only and can escape a microbial reactor, so you do not have to separate any end products. And methane as natural gas is a great energy storage. So in fact, with this system, you can store electricity outside of batteries as natural gas, which is, I think, totally cool. So we then decided to take this apart and used an organism, Methanococcus maripaludis, so in a defined system where we can do biochemistry and can do genetics, to ask the question, what is this interface of the electrode with the organism and between the electrode and the organism? The argument was always that the electron transfer from a cathode to a microbial cell is direct. And the reason for that was it was the exclusion of an alternative method. And that turned out to be false. And the reasoning was the following. A cathode can produce hydrogen, and these organisms use hydrogen. And if you measure the rate of hydrogen production in the absence of bacteria and compare this to the rate of methane formation, you see there's a discrepancy of an order of magnitude. The conclusion was, aha, and therefore there must be a direct mechanism of electron uptake. So it was essentially an argument for a rate argument that argued for a direct transfer. The alternative was that on a cathode, hydrogen is abiotically produced that is then metabolized by these organisms. So we set out to test this hypothesis. And when you take the organism, Methanococcus maripaludis, put it in our electrochemical reactor, you have a nice stable rate of methane formation. We can do this for weeks, and the rate is pretty stable. We have a nice stable current here, which draws basically electricity out. This is the abiotic system. So the system behaves nicely that electrons from a cathode are utilized and metabolized. We also saw that hydrogen is formed abiotically. So if you take just the medium without the cells, the medium which has not seen cells before, and measure hydrogen formation, you do have a rate of hydrogen formation that's like this. With cells, hydrogen is a little bit lower, and we know cells can utilize hydrogen, so we thought, okay, this is actually a result of that. We also found, and that's important from another aspect, that the same mechanism can lead to the corrosion of zero-valent iron. And you're all aware of the problems that are associated with iron corrosion. So if you take zero-valent iron granules, and then incubate this with the cells with the same medium that otherwise would oxidize a cathode, you can corrode iron and convert this to methane. And the redox potential here under standard state conditions is in the range of hydrogen that is produced at minus 420 millivolts. So with the same system, we can corrode zero-valent iron with methanogens. The system is stable, so we can essentially exchange the medium, and immediately the rate of cathode oxidation or cathode oxidation resumes, which basically means we are looking at an activity that is associated to the surface of the cathode. So the transfer really suggested that we have something direct that is linking the cells and the metabolism. So we then went into, well, I should go back. So that all looks great like direct, but we essentially haven't made any progress compared to the studies that have been done before, in the sense that we really didn't rule out hydrogen. And to do this clean, you have to have genetic knockout mutants. So we went into the metabolism of methanogens, and this is not a test. So this is essentially how organisms convert CO2 to methane. There are a number of steps in there. All in what is indicated here in red are hydrogenases. They have different hydrogenases that use hydrogen normally as their normal business plan. And in order to show that the uptake of cathodic electrons is independent of hydrogen, we had to knock them out. So we collaborated with John Lay at the University of Washington and obtained a strain where we had deletions of all these hydrogenases and then asked the question, is this strain still capable of oxidizing a cathode? And the answer was yes. Awesome. So this is the delta 6 means that all 6 hydrogenases are deleted. The strain produces nicely and stably methane. The rate was slower than we had observed before. And we also can use the same strain to corrode zero-valent iron. So while the wild type had more formate formation and hydrogen formation, I should say, than the wild type, the mutant here had a substantial rate. Now, what we also had observed is that in addition to methane, the system produced also formate. And as you can see here, formate was actually produced to a significant extent from zero-valent iron, but also hydrogen. And obviously, the mutant, which has no hydrogenase, don't produce hydrogen. So that suggested that in our system, when the cells corrode zero-valent iron, or when they are oxidizing a cathode, there is a biological production of hydrogen and formate. And hydrogen and formate can be used by the organisms and metabolized. So we suspected there might be a biological means, not an abiotic, but biological means to produce hydrogen and formate, which is then metabolized. So and that's sort of the question, is this really direct, or is there some factor being produced? So we then did another control experiment. And the typical control experiment has been, well, in order to ask the question, is something biological or abiological, you just take the buffer, the medium without the cells, and then you add cells, and then you look what happened. Since we suspected that there is some biological activity involved, we basically took a medium and simply centrifuged out the cells, and then took the cell-free supernatant as our abiotic control. So we knew there were no cells in there, but it has been in contact with biological with biology of cells. If you then take this cell-free supernatant and incubate this with a cathode at minus 600 millivolts, you get with wild-type supernatant from wild-type cells, you get formate formation. And you also get formate formation with the hydrogenase mutant. All right? You also get hydrogen. In fact, hydrogen at minus 600 millivolts here shows nicely the difference between what typically has been done as an abiotic control, which is just a medium without cells, as opposed to medium that had been in contact with cells, but you removed the cells. They produced a lot of hydrogen. So in other words, there was some factor in the supernatant of cells that enabled the formation of hydrogen and formate on the cathode, which was then metabolized. We then showed that if you measure the rate of abiotic formation of hydrogen and abiotic formation of formate, and add this rate on top of that, you can explain the rate of methane formation that we had. And that nailed it. We basically know that what looks like a direct electron uptake is actually an activity, and I can say these are enzymes. We found subsequently these are enzymes that absorb to the surface of cells, of a cathode, and then produce locally hydrogen. To really show that we have an enzyme, enzymes work reversibly. We took supernatant, absorbed it to a surface, a cathode surface, and then here ran it in a cathodic system where we had nitrogen, CO2 in the gas phase, and just looked at the current, and this is essentially a current producing a formate. We then switched the gas phase in the supernatant with hydrogen, and now we actually produced current. We don't no longer consume current because now you're pushing hydrogen through the hydrogenase activity on the cathode into, this works now as an anode. If we then switch again the atmosphere, we are back to a cathodic reaction. And then if we add formate, which we also had observed, you again push electrons into this anode, which is based on formate. If you then add everything together, it increases. So this really looks like we had a system where enzymes absorb to a cathode, locally produce hydrogen and formate that then is accessible to microorganisms. These are other control experiments that I want to skip. So one mechanism, which probably environmentally speaking for mixed cultures like you have in methanogenic complex reactors, one mechanism is really that you have enzymes sorbed to the surface that produce these intermediates that are then metabolized. Now, what is remarkable on these enzymes, and the enzymologists are jealous for that, this system, just with sorbed enzymes, is active for months. Which basically means all we have to do is change the buffer because we are running out of protons in order to run this. So there is some aspect of these enzymes that come from the Methanococcus maripaludis that makes these enzymes extremely stable and therefore extremely interesting as candidates for a sustained production of hydrogen and ferment. And I should also say there's some envy among the electrocatalyst people here because the noble metal or other surfaces from electrocatalysis lose their selectivity fairly rapidly over a course of hour or certainly a day and cannot typically withstand being in contact with microorganisms, whereas this we can run for 6 to 8 weeks. So we really need to understand the basis of that. I just want to flip to a second system that we are studying where we know the uptake of cathodic electrons is independent of enzymes to sorb. In other words, nature has several modes of taking up electrons. And this was done with a sulfate-reducing organism that was isolated more than 10 years ago based on its ability to corrode iron. And as you can see here nicely, this was in the publication of the isolating paper. In the absence of organism, this key does not corrode. In the presence of the microorganisms, this key dissolves. That's a good example for microbial corrosion. In other words, for uptake of catabolic electrons from a surface. We looked on graphite surface. When we grow this organism, there is no significant biofilms. Here you see that the organisms can associate with the graphite rod in crevices, but there is no particular biofilm that is formed on these cathodic surfaces. When the organism is sort of grown with a cathodic surface, so this is our substrate 1, the catabolic substrate, this is substrate 1, this is the substrate 2, the same as we would use oxygen. These bacteria use sulfate and produce hydrogen sulfide, they are marine. We can oxidize, and a cathode is nicely shown here, and we consume sulfate. So the organism grows on a cathodic reaction. But we also saw we're having some hydrogen formation. Once sulfate was consumed, an interesting thing happened. The current started, once sulfate was consumed, the rate of hydrogen formation in this system started to shoot up. But it didn't go on forever because actually the current sort of decreased. So we got basically hydrogen formation when the organisms didn't have any electron acceptor, and this hydrogen came from a cathode but was independent of enzymes that sorbed. The reason we were wondering why this activity stopped here is we calculated because this system ran in thermodynamic equilibrium. In other words, with a cathode potential of minus 500 millivolts and a hydrogen concentration of 10% and a pH of 7.5, this was the maximum hydrogen you can get. In other words, this microbial system could produce from a cathode hydrogen that is basically next to the equilibrium, which means it's a hydrogen-producing system that is without any overpotential. So in order to show that, we did a cyclic voltammetry. Here run at low potentials, you essentially have current consumption. If you then increase the potential here, we basically breaking here at a point which is minus 420 millivolts. and the equilibrium potential of hydrogen to protons is minus 414 millivolts. So in other words, we can push in and take out electrons without overpotential. So this, using this organism, is basically the cheapest and most efficient way to access cathodic electrons without any overpotential. We then thought, well, if this is an efficient way to produce hydrogen, can this hydrogen then be fed in our methanogen from the first set of experiments to produce methane, basically at no overpotential? And that works. Here we have the rates of hydrogen formation, and here of methane formation from with a co-culture of the sulfate reducer and Methanococcus maripaludis. The rates are basically identical. And if you then also do a cyclic voltammetry, you can show in the presence of the organism, there is no consumption of hydrogen when you put it into the system at low potential, because all the hydrogen is consumed. Whereas if you have the sulfate reducer alone, you basically can push hydrogen into the surface. Here, the hydrogen is consumed by the methanogen. So we think that this is an extremely interesting platform. This is the schematic drawing of how we push the electrons from a cathode through this strain. This does not occur via electrons directly associated with that. We are producing hydrogen, and this hydrogen is then used by a methanogen. So we have a two-organism consortium that can use cathodic electrons without any overpotential and store this energy as methane. So just to sum up, I introduced here two systems to understand how cathodic electrons can be used by microorganisms. One is an enzyme-based system, and we need to understand what is so special on these enzymes that make them so stable, much more stable than what the electrocatalyst people can produce, and highly selective. And we have a system with a sulfate reducer where we are having a direct cell contact. And we know that the organism has a periplasmic hydrogenase, and that's where we know the end point is, and we're now looking at the molecular components involved in taking up these electrons through the outer membrane and funneling them to the hydrogenase, which then releases hydrogen. I want to come back to this scheme of how we can use essentially a new type of biology, new in the sense it's not genetically engineered, it's not synthetic biology. It's a system that nature already had invented, but it's independent of sort of the classical use of biology for bioenergy, which is microbial electrosynthesis. And the reason why we are so excited about this is that we essentially can run this system just with CO2. In as much as we use oxygen, the organisms that we use CO2 and produce methane, acetate, butyrate. We can engineer several communities to make multi-carbon compounds, which electrochemically is not a feasible technology now in the near future at high selectivity. The system operates for a long time, and in a sense, this is, without going into details, the microbial, biochemical, and ecological principles are the same that operate in a sewage treatment plant. And Craig will probably agree with that. It's predictable. It's an exergonic reaction. The reaction will occur, and at high efficiency, and inevitably end up in methane, except that this methane is green because it comes from CO2. So you can store electrical energy, excess solar, excess wind, which is now becoming really prominent, outside of batteries as natural gas. You can pump methane into the gas grid for seasonal storage, for example, and use it in the winter when you need more energy. The second aspect where this platform, this type of biology is useful is to produce a group of chemical precursors that ultimately allow for the synthesis of carbon-neutral molecules. So you can produce carpets basically based on these precursors, and throw them away because the carbon comes from CO2. So depending on which way you go, you choose different organisms and optimize the system. So we are totally excited about this microbial electrosynthesis. That's my favorite slide, summarizing our collaborators and the people who did the work. The pioneering work was done by Svenja Lona, who was a postdoc in the lab and who really started to develop the defined culture system. The system was then picked up by Jörg Deutzmann, a senior postdoc in the lab, who worked out how enzymes sorb to the surface of cathodes and how this actually increases the consumption of cathodes. Frauke is a new postdoc in the lab who is working on the systems engineering aspect. Michael was a visiting postdoc who looked into more detail what actually the enzymes look like. What are these enzymes that are so stable and can sorb to a cathode? We know it's actually a complex, it's not a single enzyme, and it's way more stable than any purified enzyme that has been studied in detail. Anne is a finishing grad student who worked on the carbon monoxide aspect of this system and actually on methane formation. And Anteros is a joint shared postdoc with Tom Jaramillo's lab, where we're now looking at, in another GSAP-sponsored research, to understand, can we engineer differently the interface between a cathode and microorganisms? In other words, if we make use of the understandings of cathodic electrochemistry with new materials, can we build systems that are perhaps more efficient than the system that we described? Collaborators. Tom is great to work with. Bruce Logan, a great collaborator. We also had a joint GSAP project who really brought microbial electromethanogenesis to the system where people could study it. And John Lay, a geneticist at the University of Washington, who constructed the mutants. Again, funding. GSAP has been an instrumental player with that. We now also have funding from SLAC in LDRD and follow-up funding from the Office of Naval Research for microbial electrosynthesis. So with that, thank you for your attention and be delighted to take any questions.
Thank you, Alfred. We'll just open it up for questions first from the students.
So you mentioned carbon dioxide is the main inlet to the system. I was wondering if it would be at all possible to use it in conjunction with the carbon capture as the alternative to sequestration.
Yeah, so the question was, can you use this in conjunction with carbon capture? And you should come to the lab. I have a great project for you. Yes, yes. We have been. We submitted one to the Precourt Institute to see if we can actually use this to capture dilute carbon. And we have some totally exciting ideas. So yes, I think this is exactly the way how to think about it.
What's your rate limiting step? Is it the carbon flux or is it something else?
Well, we don't know without sort of giving away what we want to propose to do. We just don't know how it kinetically balances. Okay. But we know in the system that we have no accumulation of hydrogen or so. So it's not really the cathodic part that is rate limiting. Great question. Yes?
Could you contrast these microbial electrosynthesis systems with like other thermochemical or electrochemical methods for CO2 reduction?
Yes, so the question is, how does this system compare to other sort of chemical systems for CO2 reduction? What you have here is high selectivity. There's only one compound that is produced, and that is based on the organisms that you choose or engineer or whatever. We have high efficiency. The Coulombic efficiency is up to 90%. And the system is robust in the sense that it can operate for weeks and months. The question is, though, the rates. How can we get up rates? But it then becomes an engineering question in terms of rates versus selectivity. So it's a great question, and I think this is an important research question to put this together. Yeah.
Any other students? Yep.
Yeah, I recently went to a talk by someone who's helping with Bill Gates' new energy investment fund, and I asked about carbon removal technologies, and he said, we're not even interested in investing. It's not thermodynamically possible. I'm not a chemical engineer, but what would your response be?
Well, this is a great question. So let me see if I can rephrase this accurately. What is the efficiency and viability to use this essentially as some kind of basis for carbon capture in a decentralized third world setting? Help me if this is correct. I'm trying to rephrase your question.
And I guess I asked about kind of a broad range of carbon dioxide removal technologies, ranging from biomass energy, CCS, stuff in there about like algae and things like that. And he was like,
Yeah, I think you mentioned whether it's thermodynamically possible.
yes,
right?
It's
an uphill reaction.
Yes. So this technology costs energy. So using biomass, yes, you can remove carbon, but it's really expensive. It's inefficient, and the drain on resources, water, is huge. If you want to grow 1 ton of biomass, you need 1,000 tons of water. So yes, you can capture carbon that way, and ecologically it makes sense, but not from a technical point of view. Our system costs energy. But this energy can come from renewables, from solar. So we approached PG&E. We thought, we have this great technology here. Hey, we can save you energy, from renewables. PG&E said, well, we don't care. Even with negative energy prices, we just let it go. Southern California Gas, they love it, and we are working with them because they understand there's actually a market there. If you can capture excess electrical energy, especially from wind, there is a market there that justifies capital cost. So I would argue it depends on the setting where, although it costs energy, this can be offset essentially by storing electricity.
So let's open it up to other questions. Yes?
Kind of related to your comment you just made about Southern California Edison. So on a scale basis, if you were to put a ton of input into your reactor, how much methane would you get out?
Oh, a ton of...
Yeah, how many cubic feet of methane?
As I said, the Coulombic efficiency is 90%. So 90% of the CO2 that you would put into the system, you get out as methane. Now, we have not optimized the system in terms of biomass, how much biomass we have, what the cathodic surface is, and so on and so forth. The CO2 right now comes from Prax air, right? So we are not accessing natural sources of CO2, but we want to go there. So that we need these numbers in order to come to specific values. Also, in terms.
So in other words, if I figure out how much 90% of a ton is, how many cubic feet of methane that is, that's what I'm going to be getting.
Yeah, I can make this calculation. Yeah.
Any other questions? Yep.
I'm not related to that. Like you said, it's 90% efficiency for the CO2, but like the energy, how much energy do you if you put in this much electricity, how much gas do you get, and if you get it back to electricity, how much do you lose on that project?
So I was talking about two efficiencies. The 90% is a Coulombic efficiency, where your electrons go and your carbon. The thermodynamic efficiency is of the microorganisms is around 60–70%. Having said that, we are realizing that while we have been focusing on the cathode, the anode also needs research. We need to build better anodes. So that while we can now access cathodic electrons without overpotential, the whole cell potential, depending on how you run it, is still like 1.5 volts or even 2 volts. And that's a question of the anode that needs engineering.
Actually, on a similar line, if you just do electrochemistry for water splitting, hydrogen and oxygen production, are there lessons learned or enzymes from biology, if you forget the organism for the time being, just the enzymes, could you use enzymes for both the hydrogen evolution and, more importantly, the oxygen evolution that you can find in biology that's better than what we use today?
For hydrogen evolution, yes, that's basically what we have the system. In fact, that's because it's so stable.
Right. That's the easy part.
That's
the oxygen part.
The oxygen part is not obvious because the only real system for oxygen evolution, for the system 2, is a fairly complex that runs essentially at a potential, chemically speaking, of more than a volt to oxidize, to strip the electrons from oxygen. There is no good biological information that chemists could use. I think that's what you're thinking of. Yeah.
I'm sorry, you had a question. Yeah.
So, talking about engineering, can you talk about engineering the microbes, and does cell-free synthetic biology ever play into that? Does it give you tools that are useful?
Yeah. So the question is, what role does synthetic biology or engineering organisms play here? I would say the organisms that we have are brilliant already, and I wouldn't know where to engineer it. In particular, they are robust, and a lot of the synthetic biology misses an important point in biology, which is the robustness and the redundancy and low efficiency in photosynthesis. Engineers don't like this. Biology loves it because it's not about being good at one thing for a long period of time. It's all about adaptation. So things change. And the organisms that we have, that's why I made this analogy to a sewage treatment plant, behave similarly like that. You cannot optimize a sewage treatment plant with engineering organisms. You can engineer organisms, but they will evolve back to those that are existing already.
Any other question? Yep.
Yeah, you mentioned a couple of times these systems can run for weeks to months. Is there some kind of a limit to how long they can operate?
Proton limitation, CO2 limitation. That's what we found. So they stopped working, and then we add more protons, so change the buffer, and then they keep working at the same rate. We don't understand enough about CO2 limitation. That is part of a research question that we have right now.
Question?
What sort of concentrations of CO2 appear to be optimal?
Excellent question. What concentration of CO2 is optimal? So when you grow them in the lab, you have a gas mixture of 20% CO2, 80% hydrogen, which translates to a concentration of 20, 30 millimolar bicarbonate. So at pH 7, it's primarily bicarbonate. We found out in our studies with the combining an electrochemical with a biological system that high concentrations of CO2 can be toxic, which nobody had found ever before. So we need to understand this a little bit more, but this was an unexpected finding. I would argue you can control the level of CO2 that you put in your system. Again, even if you're coming from a point source like a cement plant or an anaerobic digester, where you use this technology to upgrade the natural gas, which is 50% CO2, 50% methane, you have anaerobic gas, 50% CO2, low-hanging fruit to upgrade this with this technology to 100% methane. I would argue you can control the amount of CO2. So practically speaking, CO2 toxicity is not an issue. In any case, most of the carbon that you have is in form of bicarbonate at physiological pHs. Does that answer your question?
I have a follow-up, a slightly different question actually. You mentioned as part of your products, you're producing methane.
Yes.
And you're producing, ethanol, C2, C4, etc. How specific? I mean, you can't beat biology in specificity. So how come you're producing 4 different compounds? Can you make it specific? I want C10.
Yes.
And what would be the performance? And would you need genetic sort of manipulation of that?
No, it's the choice of organisms. So I didn't go into the detail. Fascinating piece of microbiology and microbial chemistry. If you want methane, we have organisms off the shelf that produce 100% methane. If you want ethanol, we're taking different organisms that produce ethanol. If you want to get a C4 or actually C6, which is the interesting part, we're taking 2 or 3 different organisms that ultimately always feed off of cathodic electrons and CO2, but we're making use of a naturally occurring food chain, again, down a thermodynamic gradient to produce these end products.
Very cool. Very good. Well, let's thank Alfred again.
Thanks.